So, good afternoon, and welcome everyone to the last European Society of Endocrinology Spotlight on Science of the year, which is dedicated to hormone receptors and the modulation of ovary function. This seminar, as all of them during the year, has been sponsored by Recordati Rare Diseases. So we want to thank them for supporting this activity of the society. Remember that we will have time after the presentation for questions and discussion with the speaker. So remember that if you want to do any question to the speaker, you have the Q&A button that you can use and write your question that we will be reading at the end of the presentation. And if you have any technical questions, please use the chat box that you can find below in your screen. This session is being recorded, so you will have also it available on demand on the society websites in 3 months or so. Today is a great pleasure for me to have with us Dr. Livio Casarini. He is PhD in evolutionary biology. He is associate professor of molecular and translational endocrinology at the University of Modena and Reggio Emilia. He is the leader of a research team that works in basic and translational endocrinology, where he and his team study the endocrine regulation of human reproduction, glycoproteins, sex steroids, and the receptors. His main scientific achievements account for studies on gonadotropin-mediated signaling, GPCR heterodimerization, and allosteric modulation of hormone-induced intracellular signals. For this reason, he is the perfect speaker to talk about the functional role of FSH receptor oligomerisation in young and aging ovaries. Dr. Livio Casarini, thank you so much for accepting our invitation. And if you are ready, the floor is yours.
Thank you for the invitation, first of all. It's a great pleasure for me to be here and show my results and talk about the functional role of FSH receptor oligomerization in the young and aging ovaries. Before to start with the talk, I would like to introduce a bit my group and my city where I work. I don't know if you ever heard about Modena, that where I am based. Modena is a nice city in the northern Italy, close to Milan and Florence. As you can see by the picture, you can see the UNESCO World Heritage in Modena, but also we are famous because at the base of Maserati and Ferrari. We also had a famous singer Pavarotti in the past, and a lot of very delicious dishes that I'm pretty sure you have already tasted in your life, or you should do that. Anyway, in Modena, there is also my group. Our mentor is the Professor Manuela Simoni, which is an endocrinologist, while I am leader of the in vitro translational research units, where together with other colleagues, such as the assistant professor Clara Lazzaretti, we study the cell response to gonadotropins, but also we are have a clinical personnel dedicated to patient care and also to clinical research. They are all together here. Well, in our job, we study the cell response to the gonadotropins in the context of fertility. Before to start and go through the core of this presentation, I need to introduce a bit the arguments. This is the human menstrual cycle, in which there are two gonadotropins, the FSH and the LH, that modulate and support the recruitment, the selection, and the growth of the dominant ovarian follicles. The two gonadotropins act during the antral stage of the folliculogenesis that culminates with the ovulation of the dominant follicles in the fallopian tubes. What happened later is that the progesterone is produced upon the stimulus of a third gonadotropins, that is the human chorionic gonadotropins, that is made to produce these steroids and support the pregnancy. But, well, this is quite complex system that regulates the gametogenesis in the female and support the pregnancy. And there are several variables that we have to consider to understand how it works. One of the first variables is the diversity of the hormone produced throughout the menstrual cycle. And today we will focus on the diversity of the FSH molecules, that they plays a key role in the recruitment, in the selection and in the support of the dominant follicle growth. The FSH exists in different glycoform. As you may see here, it is composed by an alpha and a beta subunit. The beta one is deputed to the receptor binding, while the alpha has a function of transport molecule, but altogether they may be glycosylated and therefore exist in different glycoform according to the complexity of this glycan structure bound to the beta and the alpha subunit. And a rough division of this glycoform may consist in the FSH beta inhibit 24, which is the glycoform with the highest complexity of the glycan structure, while there are other two glycoform that has a lower grade of complexity. They are named 21 and 18. The name comes from the molecular weight of these molecules. Well, they have a different complexity of the glycan structure and also a different action on the receptor. And we will show better this issue in the next slides throughout this presentation. So there are different glycoforms that in nature they are produced in different amount throughout the woman fertile lifespan. So what I mean is that when the woman is at the younger age, between around 20 until 30 years old of age, the predominant glycoforms are those with lower complexity. I mean the 21 and the 18 glycoforms. While the age increases, these glycoforms with poor complexity decrease and are replaced by a higher amount of high complexity glycoform, the FSH24, which increases with the age of the women. So this is a first grade of complexity of the system that we have to understand and know before to explore how the menstrual cycle works. Another grade of complexity is due by the expression of the receptors. The gonadotropin receptors has a very dynamic expression levels during the antral stage of the folliculogenesis. And if we focus to this slide, we may see that together with the FSH level and the LH level, there is a variation also in the FSH receptor expression level, which is highly expressed at the first stage of the antral stage, and then it decreases going towards the ovulation. Instead, it is replaced by the expression of another receptor, which is the LHCG receptor, which increase in its expression and achieve the maximal level during the ovulation, and also in the luteal phase that is not shown here. Well, checking what happened according to the expression level of those receptors, we may divide this section in three different stages according to the physiological variation of the follicular stage. So at the very early antral stage, or we may say the secondary to early antral transition, the FSH receptor is poorly expressed, but start to increase its expression level. And this is the stage in which the secondary follicle are recruited and enter into the antral stage. Later, the FSH receptor is highly expressed, and this is the time in which there is the maximal selective pressure on the follicles, and it is also the period in which the dominant follicle is selected in the human species, which is a monoovulatory species. Later, the FSH receptor expression decreases, and is the time in which the dominant follicle starts the final stages of its growth and maturation before the ovulation. At this stage, the FSH receptor, as told before, is replaced by the LH receptor. So, trying to rethinking at what we know about the classical knowledge from endocrinology, in which the dominant follicle should be selected thanks to the decline of the FSH level, I think that we may change our view on these processes and focus instead on the receptors. What happened here is that the maximal selective pressure occurs when the receptor is highly expressed. So it means that in my mind, the FSH receptor could act as a selector of the dominant follicles, and in this case, it could have a dual action. The first action is those by which the FSH receptor acts as a recruiter for the late secondary through the early antral follicles. And in this case, it should activate a proliferative signal. But increasing the expression level of the receptor, the follicle enters in the selection stage in which the dominant follicle is selected. And so it means that FSH receptor may kill the cells in some way. I have achieved this conclusion after some experiments that come from the past decades, where we tried repeatedly to select cell line, a human granulosa immortalized cell line named AGL5, which permanently should overexpress the FSH receptor and in another batch the LH receptor. Well, what we have obtained is that we have repeatedly failed to obtain the FSH receptor resistant clone. What happened indeed is that while the LH receptor clones that are indicated here by the blue column start to grow and continue to grow, and we have to repeatedly and weekly dilute them in the culture to avoid the confluencing. That was not with the FSH receptor clone, that after the first stage of growth, after 2 months, this colony was repeatedly lost. So what happened during the stage in which the FSH receptor clones start to die was that they were characterized by overall high intracellular cyclic AMP levels, and downstream also by relatively high progesterone production, even in the absence of the ligand FSH. So it means that in our mind, FSH receptor in those models that permanently overexpress this receptor killed the cells. And our suspects were confirmed by the analysis of the procaspase-3 cleavage, which they are active, those enzymes that cut the DNA and kill the cells, only in the FSH receptor resistant clone. So it looks like if the FSH receptor may be overexpressed only transitorily in nature, and this is exactly what happened during the folliculogenesis, because it is replaced by the LH receptor in nature. And when we try to overexpress him for longer time, the cell start to die, and it is an apoptotic process mediated maybe by cyclic AMP. To say whole, this is not our discovery, because in the past, other groups have already observed the cyclic AMP and FSH-induced cell death of granulosa cells, at least in vitro. And what happened is that the cells exposed to high level of cyclic AMP start to become apoptotic, undergo to the formation of intracellular blebs containing steroids, but at the same time, the cells start to die. So to explain better with a cartoon this concept, a cells that overexpress permanently the FSH receptor is very sensitive to the stimulation of this receptor by FSH. They activate the G-alpha S protein, then the cyclic AMP. And the cyclic AMP is very important because in nature it is required to activate the steroidogenesis, and the sex steroids are very important to support the dominant follicle growth. But at the same time, the most steroidogenic cells start to die, and it is an apoptotic cell death. It means that the FSH receptor could be pro-apoptotic. And we have performed some experiment to evaluate how the receptor expression level could kill the cells. We have seen that when the FSH receptor is expressed at a poor level, there is a competition among several G protein, the GS, GQ, GE, and also with beta-arrestin to couple to FSH receptor. But under this condition, the FSH receptor has not a preferential cyclic AMP signaling. It induces life signal, cell survival, and proliferative signal through the activation of ERK1/2 and other proliferative signaling. When the receptor starts to be overexpressed, what happens is that this coupling curve demonstrates that the preferential coupling is between FSH receptor and the G-alpha S protein, that is the activator of the cyclic AMP and downstream to the steroidogenesis. But at the same time, there are apoptotic signals that are activated in the same cells. So one may say that the FSH receptor should kill all the cells in nature if it has a pro-apoptotic potential, but it is not. In the nature, in vivo, it is not, because the cyclic AMP signaling in vivo, where there is the system provided by TECA and granulosa cells, the cyclic AMP signaling is translated in steroidogenic signalings. And the steroids, especially the estrogens, are potent anti-apoptotic and proliferative effectors. In this experiment, indeed, we have exposed human primary granulosa cells obtained from women undergoing oocyte retrieval for assisted reproduction. We have exposed these cells to a cyclic AMP analog that cannot be metabolized by the cells, and increasing the concentration of the analogs that are the blue bars, you may see that the cell viability decreases, but the cells may be rescued in the presence of estrogens, at least within a certain concentration range. So the estrogens act as inhibitors of the cyclic AMP-induced death. We have thought to a possible model explaining what's happening in vivo according to this concept that we have developed. So there are several follicles that are attractive and are those that should have the highest FSH receptor expression. In those follicles, the FSH act as a pro-steroidogenic molecules that serve us to activate the estrogen production that should rescue the dominant follicle. And this is required to the dominant follicle to grow, but at the same time, the dominant follicle should have some characteristics that avoid the cyclic AMP-induced cell death. So we have started to think to what is the factor that may rest on the follicle instead of let him die together with all the other atretic ones. So to answer to this question, we have to gather another concept from the pharmacology by which there are some receptors that may form oligomeric structures, and it means that they cross-interact together and form a complex of receptor aggregates that respond differently to the ligand. What I mean is explained in this cartoon, in which, as you may see, there is a ligand that may act on its receptor that could be the FSH receptor and activate a specific pathway. But in a cell complex in which other G protein coupled receptor that may act as allosteric modulators are expressed in the same cells, what happens is that those molecules may interact, and under this condition, other pathways may be activated or inhibited also. So based on this concept, we have thought that the LH receptor may act as a partner receptor of the FSH receptor 1. So what should happen in the more FSH sensitive phase is that the two receptors may act together and help to bring the early antral follicle to the large antral follicle stage, where there is a higher expression of FSH receptor, but not of LH receptor. And in this case, we are in the gonadotropin-dependent stage, where there is the high selective pressure of the dominant follicles. The dominant follicles could be rescued by the replacement of the FSH receptor with the LH receptor that has a different impact on the cell metabolism. This is a theory. Another theory comes from other studies that we have performed because we have discovered that the LH receptor could not be the unique one partner or FSH receptor. There are several other G protein coupled receptor expressed on the cell surface. Another actor that I want to introduce you today is the GPER, is estrogen membrane G protein coupled receptor. At least that was the first name that the researchers that have discovered this receptor provided to this molecule, but there are doubts about its function as an estrogen receptor. In any case, it is a GPCR that is expressed on the surface of granulosa cells. It is not able to activate the cyclic AMP signaling because at the intracellular level, it is bound to an inhibitory complex made of MAGUC ACAP5 molecules that block and prevent the activation of the adenylyl cyclase and therefore of the cyclic AMP production. So we have performed some experiment and have evaluated that GPER is a very sticky receptor because when it is co-expressed with other GPCR, with a lot of other GPCR, it may form aggregates with that ones. So in the case of FSH receptor, our three-dimensional structural modeling prediction have seen that FSH receptor and GPER have a sort of affinity in the transmembrane stretches 6 and 7 that could be just opposite on the cell surface and produce the interaction between the two receptors. Of course, that was just an informatic prediction, but we have confirmed the results with the immunostaining. As you can see, the merge revealed that the two receptors are co-expressed in the cell surface and co-localized. Also, we have performed breadth experiments where we have used receptors fused with some fluorophores that we use to obtain energy transfer in the cells. And again, it is a very nice binding curve here, demonstrating the specific interaction in the surface of the cells between the FSH receptor and GPER. And also we have used the super resolution microscopy to count the aggregates of this receptor, and they form not only dimers, but also trimers, tetramers, and so on. So the GPCR world is overall skeptical about the possible formation of GPCR dimers. I mean, in class A receptor, which are the same classes FSH receptor and GPER. So to publish our experiment, we needed to provide more evidence, and also we have confirmed that the 2 receptor may form a heteromer using an antibody-based assay. It is named proximity ligation assay. Is a sort of PCR in situ, in which the 2 antibodies recognizing GPER and FSH receptor are fused with primer sequences, and by adding a sort of PCR mix, we obtain red dots where there is the proximity between the 2 receptors. This is a 5th way to confirm the possible formation of GPER and FSH receptor heteromers. So at this point, we have started to characterize the function of these 2 receptors when they are expressed together, and considering that the GPER is bound to an inhibitory complex that is bound to the GS protein coupled to the GPER, we have thought that a possible model explaining what happened in the dominant follicle is that the oligomerization between FSH receptor and GPER may block also the G-alpha S activation coupled to the FSH receptor upon FSH treatment. And to do that, we have performed a sort of displacement experiment in which the GPER and the FSH receptor and the G-alpha S protein were tagged with fluorophores inducing emitting signals that we detected with the Brett machine. And also we have increased the GPER expression level in those cells, and the signal produced by the FSH receptor and the GS coupling decreased according to the increasing levels of GPER. So it means that in some way the GPER interfered with the coupling between GS and FSH receptor, preventing maybe the activation of FSH-induced cyclic AMP production. And this is exactly what we have evaluated thanks to our assistant professor Clara Lazzaretti. We have evaluated if in cells expressing FSH receptor or co-expressing FSH receptor and GPER, the cyclic AMP is produced. And what happened is that in cells expressing only the FSH receptor and treated with FSH, the cyclic AMP increased, and it is expected. But we were surprised to see that upon FSH receptor and GPER co-expression, the cyclic AMP production was prevented, regarding the presence of estrogens, but in this case, the FSH failed to induce the cyclic AMP response in the cells. So we have thought that to induce proliferative signals, the FSH in the dominant follicle could use the beta-gamma subunits of the G protein instead of the GS, which may be remain unlocked and activate the calcium increase, AKT, and ERK1/2 pathway that are known as pathways that activates proliferative and anti-apoptotic signals. And this is exactly what we have explored with this Western blotting, in which we have observed that in cells expressing only the FSH receptor, as expected, again, the FSH induced the phosphorylation of CREB because it depends on the production of cyclic AMP. So these 2 results are strictly connected. But at the same time, in cells co-expressing FSH receptor and GPER, we did not see the CREB phosphorylation, but instead we have seen the AKT phosphorylation. So this is what happened in the model that we have thought. And this is beta-gamma dependent phosphorylation of AKT because it is inhibited by the specific beta-gamma inhibitor named gallane. As you may see, the FSH in this case is not effective in activating the AKT. And it is a nice data because the AKT is linked to the increase of cell viability under FSH receptor and GPER co-expression, but in the same model, the FSH increasing the cell viability when we add the beta-gamma intergalin to the cell culture. So we have confirmed that our model could be true and have started to rethink at the folliculogenesis, in which the FSH receptor expressed in the atretic follicle serves to produce, in our mind at least, a good estrogen level that serve to feed the dominant follicle growth, which should avoid the apoptotic death induced by the FSH receptor, thanks to the reprogramming of the FSH signals that is supported by the GPER and FSH receptor oligomerisation. So at this point, we have started to produce several other control experiments that I will not, I just mentioned to be short and simple. So, but just to say that we have done those experiments to strengthen the results. We have produced cells expressing a mutant GPER unable to form heterologous FSH receptor, and in this case, the FSH treatment decreased the cell viability and increased the cyclic AMP. Also, we have downregulated the GPER endogenous expression using a small interfering RNA probes, and also in this case, the FSH receptor induced the cell death and cyclic AMP increase. Also, we have produced an ACAP5 knockout and disrupting the inhibitory complex. GPER and FSH receptor has interacted in the cell surface, but GPER was not effective in preventing the G-alpha S activation induced by the FSH, and again, the cell start to die. So instead of talk about data from cell lines, I would prefer now to give some more indicative results of what happened in the in vivo environment. So we have used now a model that is more physiological than the HEC293 cell line. What we have used here are human primary granulosa cells, again obtained from IVF women. And also we have started to explore the expression of FSH receptor and GPER in slides of ovarian tissues. And during the antral stage, both the receptors are co-expressed in the cell surface of the dominant follicles. But we have also provided other experiments in which we have obtained granulosa cells from women undergoing assisted reproduction, and we have divided in two different groups those women. In the first group, we have defined the women as normal responders because they were good producers of ovarian follicle in response to the FSH based ovarian stimulation in vivo, while the sub-responder women were those with less than 4 follicles, that in our mind, it means that are bad producer of follicles because the response to the FSH was affected by the high pro-apoptotic potential of the ovaries. And it means that maybe the expression level of GPER and the FSH receptor in those models were different. And that was exactly the case, because in the normal responder women, which are indicated by the green lines, as you may see, there is a positive correlation between the FSH receptor gene expression level and the GPER gene expression level. So it means that more is increased the propoptotic potential induced by increasing FSH receptor expression level, increase also in those women the blockade of the pro-apoptotic effect of FSH thanks to the GPER expression, increasing expression. That was not in the sub-responder, where increasing level of FSH receptor did not correspond to the increasing of GPER gene expression. And in our mind, it means that those ovaries has a higher apoptotic potential due to the lack of proper gene expression level, and therefore they are indeed sub-responder women. The data was corroborated by another way to analyze the same data, in which we have replotted the data from these women, in which on the y-axis we have the total oocyte number, and it is plotted against a ratio between estradiol and FSH serum levels that in our mind is a measure by which the estradiol may counteract the apoptotic action delivered through the FSH receptor. And we have the same positive correlation between the normal and the sub-responder women, but not, in the normal responder women, but not in the sub-responder women. Well, it means that the FSH receptor and the GPER may form oligomers in the cell surface, but this is just one of the level of complexity that we have mentioned at the beginning of this presentation. We have not to forget that there are different glycoforms produced throughout the women's lifespan that may modulate this system. And a very nice experiment is described in another paper from colleagues from USA and Mexico, in which they have characterized the effect of these two different glycoform with different complexity. They have exposed cells expressing the FSH receptor to the low complexity glycoform, which has a higher potency in activating the receptor and also in inducing the activation of the cyclic AMP. And while the high complexity glycoform has a lower potential in activating the cyclic AMP. So it means that these two molecules may have different action according to the age of the patient. And we don't have to forget that since several here, it is a fact. It is known that the FSH receptor may form oligomers in the cell surface, and when, as explained in this coupling, in this binding experiment, in which the cells expressing FSH receptor fused with bread tags, in the absence of FSH, induce a specific signal. It means that they has a constitutive homodimerization of this receptor, but in the presence of FSH molecules, they increase the signal because start to increase the number of events in which the FSH receptor contact itself. What happened in this setting is that low glycosylated glycoform should act and induce a very good cyclic AMP production and likely a very good estrogen production, because there are data from colleagues in England that shows that those glycoform have higher propensity that the highly glycosylated glycoform in act towards receptors that are maintained as a single molecule or at least as dimers, but it is not when the cell treatment is performed using the highly glycosylated FSH molecules, the 24 glycoform, in which there is a poor cyclic AMP and also likely a poor estrogen production, because the highly glycosylated glycoform induce higher complexity of the oligomeric formation of FSH receptor, and under this condition, the cyclic AMP production is less effective. And maybe it is the explanation by which, in the previous slide, also the other researchers have observed a different cyclic AMP production according to the stimulation with different glycoforms. But under this context, and it may mean that when we have poorly glycosylated FSH glycoform, the single molecules FSH receptor are free to act and react more freely with the GPER that are expressed on the same cells, but it is not when they form more complex aggregate that one or zero single GPER molecules may counteract very weakly the activation of this cyclic MP, and therefore also the estrogen production. In this case, maybe the ratio between cyclic MP and estrogen is different, and overall it could be the setting that occurs in an ovary of a poor women of old reproductive age. They are hypotheses, of course, but it's a speculation based on the existing data. But let's see, in the next year, someone may demonstrate that it is true or not. So concluding this first part of the talk, the second one will be shorter. We may say that the FSH receptor expression level may act as a modulator of light and death signal according to the FSH receptor expression level, and the oocyte survival in the dominant follicles is determined by oligomeric formation between FSH receptor and other partners. We have identified GPERs, there is also LH receptor, but also the formation of FSH receptor oligomers may play a role according to the glycoform produced throughout the lifespan of a woman. And in my mind, it is a sort of endocrine adaptation to the aging follicle that needs different signals to avoid an excess of pro-apoptotic signaling. They are hypotheses again. But during the menstrual cycle, what happened later is that the FSH receptor is replaced by the FSH receptor during the LH-ACG dependent stage. And the LH receptor is a molecule that is very similar to the LH receptor, to the FSH receptor. Indeed, we have discovered again using a bioinformatic approach that the same transmembrane stretches of GPER react with the transmembrane structure of LH receptor, and also the proximity ligation assay and other approaches such as PALM and BRED has confirmed that GPER may act and form oligomers also with the LH receptor. So we have started to characterize also these oligomeric formation and their role in the ovarian cells using a functional experiment in which Clara have treated cells expressing LH receptor or LH receptor GPER expressing cells, but we were surprised to see that the effect of GPER was not consistent in blocking the cyclic MP induced by the LH or ACG ligand stimulation of those cells. It means that the behavior is specific for each receptor, and what we have seen in the FSH receptor expressing cell is not confirmed in LH receptor expressing cells, and therefore the role of GPER also may change during the menstrual cycle according to the stage, which could be the antral one or the luteal one. But we were also more surprised to see a specific effect in counteracting the activation of the GQ-PLC activated pathway. In this case, in the cells that co-express the LH receptor and GPER, both the ligand, LH and ACG, failed in activating IP1, which is a sort of molecular endpoint for the phospholipase C pathway. We have also demonstrated that it is the same for the calcium ion, which is another endpoint on the same pathway. These are data not shown, but you may find them in the paper that has been accepted today in endocrinology. So I hope you can check this paper. But this is a different behavior that we have thought it could be due to different transmembrane branch 6 and 7 between the LH receptor and FSH receptor. They are not identical, and maybe those variations in this amino acid location are enough to provide a different interaction and a different response. But to give a more physiological weight to this data, we have started to think if those heteromeric formation may play a role in the physiology, because we know that in human granulosa cells, the calcium increase is very important to produce steroids and during the ovulation. So we have thought that the GPER may play a role in granulosa cells at the luteal stage when the LH receptor is highly expressed. And so we have obtained human granulosa cells from at the luteal stage in women undergoing assisted reproduction again. Those cells, in those cells, again, we have confirmed that both the gonadotropin may activate the cyclic MP response and downstream the progesterone production. So even in this case, the cyclic MP production and pathway was not affected by the presence of GPER, but unfortunately, it was not for the cyclic MP IP1. So in this case, the GPER, and differently than what observed in the HEC293 cell line, the GPER failed in downregulating the production of calcium and IP1. But even more surprisingly, also the gonadotropin has failed in inducing the activation of those pathways. So it seems that in our model, in the luteal granulosa cells, at least when they are cultured in vitro, the gonadotropins cannot activate this pathway. And so this is not a proper model to study what happened in vitro between LH receptor and GPER. This is an unfortunate situation, but we have also explored better the reason why the gonadotropin failed to activate this pathway, because we have analyzed the expression level of the GS and GQ coding genes. They are GNAS and GQ, GNIQ. As you may see here, when in the transfected HEC293 cell, there is a very high expression of LH receptor, lower expression of the GS protein, and also a poor expression of the GQ. But anyway, this poor expression is anyway enough to induce a coupling with the LH receptor that may interact with both GQ and GS and activate both the cyclic AMP and the IP1 pathway. This is not in the human primary granulosa lutein cells, where the LH receptor is poorly expressed compared to the GS protein, and maybe the preferential coupling is between LH receptor and with the excess of GS protein, therefore omitting the coupling with the GQ. And under this condition, the granulosa cells and gonadotropin failed in activating the phospholipase C pathway, and so the IP1 and cyclic AMP molecules. Indeed, we have repeated those experiments in cells, in granulosa lutein cells, transfected with GQ coding plasmid, and in this case, the gonadotropins activated the phospholipase C response. So we have obtained from this data, from the study of LH receptor and GPER oligomerisation, this concept by which the LH receptor, according to its expression level, may lead to a preferential coupling to a GS protein, but when it is poorly expressed, there is only the GS coupling, while not the GQ coupling, and therefore only the cyclic MP production. So to conclude this presentation, we may say that after those studies, we have understood that FSH receptor and GPER oligomers seems to have a physiological role, but it is not for LH receptor and GPER oligomers that needs further confirmation, maybe in granulosa cells obtained from the antral stage. So there is a problem in the cell model. Anyway, I hope that I have convinced you that the oligomeric formation of FSH receptor and other partner receptor expressed in the same system may have a role in the human physiology. I conclude with the salutation of my collaborators and supporters. I have already introduced it to you my group, so now I would like to thank my colleagues, Eileen Hanialoglu at the Imperial College London, Adolfo Rivello Müller, and all other friends that are listed here, as Eric Reiter and Francesca Fanelli Meili. And thank you also for your attention.
So thank you very much, Dr. Casarini, for this great presentation. I think you made an excellent overview of the role of FSH receptor and the physiology, oligomerisation, and interaction with other receptors with ovary function. Now, as we mentioned at the beginning, we have time for question and discussion. Remember that all the attendees can use the Q&A button to send questions for the speakers. So, and indeed, we already received one. It's quite clinical, but I think could be of interest. I think Livia, you can also read it on the Q&A.
Yeah,
but
yeah.
the question is, well, thank you very much for your interesting lecture. I have one patient, a 15 years old girl with FSH receptor gene homozygous mutation, plus chronic kidney disease. I read some paper that very elevated FSH in menopausal women can bring to kidney disease and fibrosis by catenin pathway. Your opinion is very interesting if you can say something about that.
Well, to be honest, I don't know if there is a link between the FSH role and the kidney, because my knowledge relies on the fact that I strongly believe that there are no extragonadal FSH receptor expressed in the human bodies, at least in the physiological condition. This is not in the pathological condition in which tumor cells may express them, but in the physiological condition, there are no extragonadal FSH receptor, and therefore, in that patient, I don't know if the FSH act on the gonads and some steroids or other molecules is produced and act as a mediator of the FSH action, indirect action on the kidney. I think that the two things are not related to each other, but I admit that I'm not expert in the kidney disease.
Okay. We have another question related to the dimerization of oligomers, GPCR oligomers. Very fascinating work and presentation. Is GPCR dimerization or oligomerization mostly depending on raw co-expression levels in the cell membrane? Or is it rather induced by ligand binding and receptor activation, or a mix of both of them? What do you think?
Yeah, thank you also for this nice question. There are several experiments that has demonstrated that the oligomerization or dimerization is dependent on occurs also at the constitutive levels. It means that in the absence of a ligand, those receptors may interact. The ligand may stimulate the production of more complex oligomeric structure, or also induce the dimerization. This is already demonstrated. But the main issue here is that I'm strongly convinced that it depends on the expression level of the GPCR. When they are poorly expressed, I think that the oligomeric events are quite rare in the cell membrane. So only in specific physiological moments in which one specific GPCR is highly expressed, and it is the case of FSH receptor during the antral stage. Well, in that case, when the receptor expression achieves very high level, there is the possibility that it interacts with other receptor at the physiological meaningful level.
Mm-hmm. Okay. I wanted to ask you also, because you have shown that this FSH receptor can interact with GPR13, and it's quite clear, but do you know if there is interaction with other GPCR? For making a more broad question is, do you know if we have now some technique that can allow us to determine which are the receptor with one specific receptor is interacting, rather than testing if this one is interacting with this one or so?
Yeah, thank you for this question. Well, this is the main limitation in the study of GPCR oligomerization. We lack of proper model, and also it's very hard to study receptor oligomerization in vivo in the physiological context. So we have to use cell lines that are subjected to produce very often artifacts, because we act under high FSH or other GPCR overexpression, and in that case, our handling induce in a certain way an artificial contact between those receptors. We have to ensure under different conditions if it is the case also at the physiological levels, and what we have in our hands are super resolution microscopy that we may apply in cells, in primary cells obtained from original tissues. But they are antibody-based method, and so we have to use reliable antibodies, and this is another limitation. Yeah, especially in the world of gonadotropins, there are a lot of papers detecting extragonadal receptor, but this is one of the main limitations. Well, that's it. So we need several approaches to demonstrate that a cell model is reliable, and the two receptor may really interact, as you have seen in the presentation. The reviewer asked for us to additional method, not only the bioinformatic approach, not only the immunostaining, but also breadth, proximity ligation assay, and so on, to check if it is really true or not.
Yeah, we know how difficult is working with GPCRs, right? So I think, but you so clearly, which is the role of FSH receptor and the oligomerization with other receptor in the physiology. So I think it was a great presentation, great work. I think it was a really interesting session. So we don't have more questions from the audience. So I think it's 7. We can close the session again. Thank you so much for accepting the invitation to be here. This is the last Spotlight on Science of this year, so we will see you next year with a new series of Spotlight of Science. So thank you so much, Livio, again.
Thank you. Thank you too.
Have a good one. Bye-bye.
Bye. Thank you.